So this is going to be a brief sort of introduction to pharmacological fMRI, along with sort of an adjunct that you can do when you're doing drug work, which is add things like EEG fMRI as well. So I'm going to start with describing what pharmacological fMRI is. It's pretty straightforward. The reason that it sort of becomes, it could, that it could become very important and quite lucrative in the future. The different types of studies that you can do, a couple of examples, some of the confounding factors of using fMRI with this kind of, with drugs, and how to maybe control for those kinds of factors. And then go into some basics of EEG fMRI, how to do it, and some of the issues with it, and then a couple of examples. So pharmacological fMRI is pretty straightforward. It's an fMRI experiment where you add a drug. And you can either look at the drug activity itself, so how it just modulates sort of the baseline of the brain images, how it modifies the task activity, so you can either give the drug beforehand and then see what effects it has on the task, or during resting state as well, to see whether it has any effects on the resting state networks. So one thing to keep in mind is that fMRI, as you've heard in a lot of different previous lectures, I'm sure, is derived from a blood oxygenation level dependent signal. And so that signal is a function of a couple of different types of changes, metabolic oxygen consumption, or the rate of signaling, the blood flow changes, and blood volume. So all of these things can be modulated by drugs if you're giving them. And also the vascular coupling between the neural signaling and the blood flow will also change, and it changes with age, could change in disease. So the points of doing, the reasons to do pharmacological imaging are obviously to try to demonstrate what effect the drug has. Does it actually get into the brain at all? What type of dosages are going to give you the results that you're looking for? And to also in development of the drugs, to give to give companies go-no-go decisions in drug development, because as I'll describe in a second, it's actually quite hard to come up with new compounds and actually, ones that they don't have harmful side effects and actually do what you want. One thing that's that imaging is supposed to be, is very promising for, is to actually directly identify the targets. So historically, the drugs are not developed, at least for looking at the brain, they don't know exactly what brain region those drugs are going to target. So if you do imaging and you know that a particular disease has a dysfunction in the insula, you can then maybe try to design your drug to actually target the insula directly. And so I was sort of alluding to that point in terms of looking at the mechanism of action of the drug in the brain. Furthermore, instead of just doing paradigms to manipulate or to give people paradigms to do and have a look at their responses and look at the networks that are involved, you can actually give people drugs to actually see if you can modulate things like their consciousness level. Usually we do that for sedation, but you can actually check to see what the brain systems are being used for. So drug development is a really long process. If you look at this slide if you look at it looks like it takes about 15 years to go from start to finish. You start with, say, 10,000 candidate drug compounds, and you whittle it down over a number of years. Either they don't actually reach their targets, or they're toxic to the people, or they don't really work in subgroups, and so on and so forth. So you can see that the number of drug approvals have decreased considerably. In fact, there's only basically 22 novel compounds that were approved last year. But conversely, the amount of money that's been spent on prescription drugs is going up. So there's clearly a need to make this a more rapid process, because there's a need for new drugs to treat different diseases and so on. But a lot of companies have actually given up, particularly in CNS drugs, or a lot of companies have given up developing them because it's so costly to do it, and that you basically don't get very good return on your research money. So that's where imaging might be able to step in. Because a lot of the clinical trials basically fail, you might want to actually put imaging in early in the process so that you can identify directly the target, so that you know that the drug is going to actually work before you actually put it forward to the further phases of the clinical trial. And trying to identify, you also might be able to identify subgroups of patients that might actually benefit from the drug, rather than having to try it across like thousands and thousands of people. You might find a couple of hundred people that it might actually work for. So across each of the phases, just to be explicit, there's four or five different phases of a clinical trial. So the first one basically looks at the safety or tolerability. So in terms of the use of imaging there, it's basically just looking at the pharmacodynamics of the drug. In phase 2, which is still several thousands of people, basically, or not is it scales up. So it's only a couple of hundred people. You're looking at trying to make sure that the drug actually works very well, so you can actually see if the drug is actually hitting its target or not, and so you can make a decision as to whether the drug can go forward to further trials in humans. I'll talk a little bit about trial design in a second as well. So post actually determining that the drug will actually work, say you're trying to market the drug to other people, you can potentially then have a look at subgroups of people, again, to make sure that it works, and actually directly market the drug to people where it will work, so that you don't have to try multiple different drugs as they do right now in depression, and still have multiple failures, so the person will actually get a drug that actually works for them. So these are basically all ways in which imaging can try to reduce the cost of bringing a drug to market. So one of the special things about the brain is that it's got the blood-brain barrier, so not all drugs actually can reach the neural targets. But since fMRI in particular is non-invasive, you can actually see whether or not it's actually modulating any of the brain systems. Of course, there's a slight confound with fMRI. You're not sure that it's exactly doing something at the neural level. But nevertheless, you can at least see whether or not there's a change in the patterns of activity while the drug is being given. And not only that, you can measure it at multiple different time points because it's non-invasive. And because it's non-invasive, you can actually do this in healthy controls as well, instead of just only focusing on diseased populations. And then you can follow people for longer periods of time as well. Another important thing is that it's an adjunct to subjective responses. A lot of the CNS drugs, say for depression, rely on clinical scales, so people actually self-report whether or not they're feeling better on the drug. And of course, something that's pretty well known is the placebo effect, so they could just think that they're feeling better, but they're not actually feeling better. So using imaging can actually give you an idea as to whether, like an objective response, or an objective way to actually see whether or not they're actually responding to the drug or not, or they're getting a reduced effect, or actually they're not getting any effect at all. So the different types of studies that you can do, it's reasonably obvious. It doesn't really matter what time is on the x-axis. But if you've got a drug and it has a particular effect, and then the drug effect turns on, and then it has a washout period, what's popular to do is a task beforehand. You give the drug. You might try to actually see what is happening at the acute phase, and then also if there's any activity that's changing during the washout period. If you've got a short enough a drug which has a short mechanism of action, you can actually do the image during the whole thing. And usually what happens is you can't actually image during the whole thing. You might have things like IRB requirements of making sure the person's blood pressure and heart rate and so on hasn't changed while you're giving it, so you might have to break up your imaging periods into several different shorter time periods. So all of these things have pros and cons, but one of the things that is a lot of the studies are basically done pre and post drugs. So I'll just give you a couple of different examples. one in resting state where they've given them a drug called midazolam. It's a drug that's used in sedation, also used, I think, in maybe epilepsy. And they were just looking at resting state networks. And you've had, I think, a whole talk on resting state networks. And so they were looking at the effects of this drug on the motor network and the default mode network. So during sedation, I think they gave them the drug beforehand, and then they gave them a scan afterwards. And you can see that the networks are actually still there, but they're slightly changed in only one or two different locations. So they were pleased to see that the resting state networks actually persisted when the person was mildly sedated. There's also, this is also very interesting, like pain at least is is a topic that a lot of people have done research in with this kind of thing. So here on the top graph, you actually see during each of the dotted lines, there's thermal stimulus that's being applied to the back of the person's hand. So they get basically heat stimulus every like 25 seconds. So that's basically what's happening in the first graph. In the second graph, they're actually given a pain drug, which is called remifentanil. And you can see the dosage of it increases and then wanes. And as the dosage is increased, you can see that there's a decrease in the pain, in the response to this particular pain stimulus as well. And the people were also reporting a decrease in sensation of pain. So you can actually see that doing this kind of fMRI task, you can actually sort of directly measure the reduction in pain that the person may be experiencing. The other thing you can do in this particular case is looking at the acute effects of drugs. So this is nicotine, which is being injected. So they get a 1-minute bolus of nicotine that's injected here. So before that is baseline, and you can see there's a really quick response to nicotine, and so people would feel more relaxed and so on. But it wouldn't last very long. And in this particular study, you can actually also basically look at the different dosages because it goes up and down so quickly. And correlate this kind of response to like blood serum response of nicotine. And the last example that I'm going to give it's very similar to the previous example, but what they're trying to actually do here, it's again with the pain drug, is to look at the pre-infusion state and look at the pharmacokinetic response. So actually try to model the drug response in the brain, which may be different from any of the peripheral measures that that you would get from just taking blood draws. So all of this is basically just to say that you can do this. This is not an exhaustive list of examples. I would refer you to this paper, which is a pretty good summary of a lot of the resting state types of experiments that have been done. You can see that a lot of different drugs have been tried over a bunch of different CNS modulating things in terms of like looking at different receptors and for different reasons as well. So either looking at the effects of the drug or trying to actually just figure out the doses, or clinical things like basically taking somebody who's depressed, giving them a drug and seeing if you can see any changes in their various networks. So as I mentioned before, just doing fMRI might not be enough in terms of looking at the effects of the drug because you're not actually getting a direct neural measurement. And I'm going to go through 3 different confounding factors. It's probably not an exhaustive list, but gets you at least started in thinking about this. I sort of briefly alluded to cognitive things that might affect the response to the drug, which is one of which is the placebo effect. And I'll talk about this one next in terms of how to look at that and fix it using maybe study design. I'll look at some acquisition effects, just because the drug effects are slow and are on the same order as some of the artifacts in fMRI, and then some of the signal changes that I've been mentioning also. So if we start with the placebo effect, I think it's pretty well known what that is. It's sort of driven by the expectation that you're actually going to get a treatment. And it's actually pretty interesting. I mean, historically, this was actually found, I think, in World War I or World War II. I can't remember exactly when morphine was actually used in the field. they had run out of morphine basically, and they were doing surgeries on people. And one of the nurses was like, "Well, I don't have any morphine. I'm just going to give the person saline." And lo and behold, the person actually thought that they had less pain. And so the surgeon who was in charge then went back and sort of started looking at the placebo effect. So if you give somebody an injection, they'll feel better than if you give them a pill, than if you give them various things. And it even comes down to just the color of the pill as well. But not just those neural effects that you see. Like, it's not just behavioral. People have actually shown, in terms of the brain networks that are involved, in this case, you're seeing regions that basically are responding to an antidepressant drug, and they've also done scans in the placebo condition and with the drug. So any of the regions that are filled in are regions where the placebo condition and the drug basically are affecting them at the same time, or both of those regions are affected by both of those things, and in both in the positive and also in the negative. There are, of course, a couple of regions that are unique to the drug, like the basal ganglia, the anterior cingulate, the anterior insula, and the hippocampus. And then there's regions that are, one region, I guess, which is unique to just the placebo condition. So if you're trying to isolate drug response, it's going to be important to try to control for this. And so the standard way of doing that is basically by giving both the drug and the placebo. So that comes down to study design. You can either take all of your people and split them into giving them different treatments. Of course, that's going to require more people. And also then you have to account for the differences between all of the people, because not everybody responds the same way. So if you want to try to control for that, you can do something called a crossover trial, which means the same person goes through both two arms of a study. They either first get the drug, you wait for the drug to wash out, you then give them the placebo, or the converse, the person, so the same person either will get the placebo first or the drug first. In some cases, it's sort of unethical to, say, maybe take the person off of medication, which is sort of saving their life. So you can't just have a clean sample. You can't just take the person off the drug and then give them a different drug. So you find people who are matched, so they are either taking the same drug, and then you then try to give them an adjunct or the placebo. So in terms of these study designs, you actually then want to think about whether you're giving an open label drug, whether you randomize it, whether both the tester and the person who's receiving it are blind to whether or not they're getting the drug, because even if the experimenter knows that the person is getting the drug, they may it may affect how they either tell the person what they're doing or not, and so then affect whether or not the person thinks that they're gonna are feeling better later. And also whether or not you want to do placebo control. The reason for in this particular case, I think I'm meaning more an active control. So there are certain drugs that are psychedelics, right? And so it's very difficult to find a drug that mimics the psychedelic, which is not actually the psychedelic itself. So you want actually an active control, because sometimes people can actually tell that they're getting the drug or tell that they're getting the saline injection. So all of these kinds of manipulations basically increases the number of subjects that you need for all of your trials. So you can see that it can rapidly get out of hand if you're trying a drug and placebo, and you've maybe got healthy volunteers and patient populations. So it really then comes down to just trying to figure out exactly what it is you want. Do you really need to know what the effect of an antidepressant is in a healthy patient, or are you really just interested in finding out what it does for just the depressed subject themselves? Also, you have to consider the reliability or repeatability of your measurements. Say you're doing multiple resting state runs in the same person. Do you know whether or not the differences that you're getting from one run to the next are because of the drug, or whether it's just sort of random variation in the person's resting state networks itself? And lastly, basically in terms of drug action, if you're doing crossover drugs, you have to pay attention to how long the drug takes to act. So some antidepressants, for example, take weeks. So this clinical trial could take months and months and months. If you're trying multiple drugs, you're taking them off the drug, you have to wait for the drug to take effect, you have to wait for the drug to stop being effective, and then give them so you can imagine that these trials can take a really long time. So just a couple of examples from the group that I'm currently in. This one just finished up, and you can see how we're sort of trying to integrate imaging into different arms of the study. So here's an example of a crossover design, and you can see the washout period is, in this case, 14 days, because it's a pretty, ketamine is a pretty rapid-acting antidepressant. And we've got, studies which actually have multiple different groups as well, and they know that they're getting treatments. We're also trying to do imaging during the infusion so you can actually see what's happening. And we also try to integrate different modalities at the same time as well. So now on to looking at acquisition issues. So if you're trying to look at, say, something during an infusion and you're trying to image something over, say, the effect takes about 10 minutes, those are really slow changes, and they're at the same scale as drift artifacts. So let me show you exactly what I mean. If you're looking at, say, bold response, a lot of the tasks are, say you have a block design, they're pretty short, they're about 30 seconds on and off. You actually get a pretty good response. And here what I'm comparing it to is arterial spin labeling. I think you also probably had a lecture on perfusion as well. So this basically doesn't have the drift artifact the same way as bold does, because it subtracts pairs of images, right? But even if you don't want to compare it to the gold standard over here, you can see what happens with the fMRI result as you sort of just increase the length of times of your blocks. So from 30 seconds to a minute to 2 minutes, it's still doing pretty well. It's a motor task, so it's activating bilaterally the motor cortex. But as you go to longer and longer time periods, you can see that the bold response sort of drops off. And it's not because the person isn't doing the task. You can see they are still doing the task. It's because the drift artifacts aren't being corrected for properly or rather they're trying to correct for the drift artifacts, and so at the same time they're removing their signal. And the reason that we might want to do bold instead of ASL is principally because it has greater signal strength, so you might be more sensitive to smaller responses. So what people have done is use different types of models to try to compensate for that, the fact that you don't actually know where your drift is. So they try to model it by doing either a linear drift, quadratic drift, so on and so forth, but you can see they're basically just guessing. Even though they still do manage to actually see some signal in all of their subjects, you're not actually sure that what they end up seeing as a result of that is really more of a function of their model. It could still be motion, so you want to actually be sure that that's what you're actually really modeling, just the drug response. So the problem is with single echo data, you're basically, you can't tell your drifts from your bold signal. So that's what people are doing. They're trying to model it. They're trying to do ICA components. They have to remove things, but they're still kind of guessing. So if you remember, basically bold bold is a difference in oxygenated signal difference between oxygenated and deoxygenated blood. With single echo data, you're basically getting that measurement at a single time point. But with improvements in hardware and software, mostly acceleration, you can actually now get images at more than one time point and still have a reasonable TR. So if you sample your signal decay at multiple time points, you can then actually measure signals, the scaling of the signal at every time point. So if your signal of interest is scaling with the echoes that you're acquiring, you know that it's a bold signal. So if it wasn't a bold signal, basically the signal would not scale. So what you're looking for are signals that scale across these three echoes and signals that don't scale. There's, I think, a talk later on, which actually will go into multi-echo acquisition in more detail. But for now, basically, you can just take away from this that it's possible to actually take an image at multiple echoes and then be able to sort which components are bold components versus components that aren't. So that way you have a more principled way of throwing away all of your drift artifacts. So let me give you an example of how this would work. And so I think people are familiar with sort of the visual stimulus, which is a flickering checkerboard. It's a little bit annoying if it's at full contrast. But you can actually reduce the contrast quite nicely. So you can manipulate it, the contrast, and manipulate your bold signal. So this is sort of you can imagine this is a surrogate of actually doing something like a sort of drug response where you get sort of a declining or a slow slope. So you can manipulate it either in a block design or with long blocks or, these nice little ramp things. So, if you sort of hypothesize what's going to happen, I'm not going to show you the block response because I think it's going to do pretty well. I'm going to show you a long block and one of these ramps. So you can imagine then that this is just the raw data for this long block where you have like high contrast here in the flickering checkerboard and low over here. You can actually see the block, but you can also see this slow drift that I've been talking about. And for the block task, you can actually see it pretty well, but it's got a little bit of drift too. But now with this ramp task, that drift basically makes it look like there's no task at all. It looks pretty flat. If you're looking at, so most people will detrend their data. You can detrend the data, you can take the trend out, so then the block looks perfectly nice. Of course, if you detrend a flat line, you end up with a flat line still. And of course, the denoising with the multi-echo works very well, so you can actually still see the block, and then you can also see the ramp task. I'm showing you the non-bold data as well, just so that you can explicitly see the ramp. And so this negative slope plus the positive slope is basically giving you this zero slope that you weren't detecting. So using multi-echo, basically, the upshot is that using that will sort of enable you to look at these slow effects. If you're just giving somebody an injection of a drug and you want to actually see and model what's going on, you can do it very directly by using multiple echoes at once. And this is just sort of a cross-verification of, not only is the time series okay, but also you're not just getting data. It's not removing signal from all over the brain and giving you just you actually just get a localized signal only in the visual cortex, which is what you expect, right? You don't expect to be able to get response all over from doing this denoising. Lastly, before I'm going to start so this is sort of one of the last components of these confounding factors. The bold signal is affected by changes in blood flow and volume. So what are you going to do about that? Well, you can use other measures, which actually maybe directly measure the neural signal. So just to remind you, bold is sort of a combination of metabolic oxygenation changes, blood flow changes, CBF changes, which gives you your change in hemoglobin. But really what we're interested in is just the neural activity, right? So if you have a look at this nice matrix, which is a little bit busy to start with, but if, say, you're looking at the baseline condition, which is, this is your standard neural response, this is your standard sort of signaling in your neurons, and your standard vascular response, you get this response for your bold, for, say, your task. But now if you give them a drug, you basically change, or if it's a different population, you've got a different neural response. You want to see that your bold response is scaling either up, in this case, or down with your neural response, and that there's no change in your vascular response, right? This is the condition that you're really looking for. Of course, what might actually happen is that you get a neural response, but you get a decrease in vascular tone, and so it looks like you've got a decrease in signal, which means you're not actually modeling this correctly. You would, from this, conclude that, well, you've got a decrease, but really, actually, you had an increase in neural response, and the converse is also true. So how do you pull out just the neural response? So you can't do that with just bold on its own, but you can do things like use either MEG or EEG, because they actually sort of look at neural activity more directly, rather than rely on vascular coupling and so on. So, if you're looking at MEG, you probably can't do MEG and MRR at the same time. They both require, large pieces of equipment, and so you're not guaranteed to get the person in the same state on both scans, right? So you basically want to do them simultaneously. So you can do EEG and fMRI simultaneously. So that's what I'm going to be talking about a little bit next. So the signal origins of EEG, basically they come from the excitatory postsynaptic potentials. It's basically summing all of them together, and that gives you your EEG waves. And they're very short. right? So they're sort of on the order of a few milliseconds or hundreds of milliseconds versus the Boole response, which is, a lot longer, sort of 5-6 seconds, and lasts for a while. So simultaneous EEG, sort of, there's technology that now enables you to do them both at the same time, and hopefully then you get both the benefits of the spatial accuracy, or the spatial resolution, rather, of fMRI, and also you can then combine that with the improved temporal resolution of EEG also. So these things are possible to combine, but it's not super common. So this is just a PubMed search of resting state in human fMRI, resting state with the EEG in the blue, and then just what happens when you look at just simultaneous EEG fMRI publications. So you can see there's this rapid drop off. It doesn't seem to be very popular, even though you might be able to get a lot of good data out of it. So this is partly because the setup is pretty complicated. You need to have some dedicated EEG equipment. You still need your stimulus computer. You still need the scanner, and you also probably are going to still acquire your physiological data. So that's 4 different computers that you have to run at the same time. You've got to make sure that your EEG is synchronized with your scanner and so on. So it's just a little bit of extra overhead. To compound that, basically, if you're looking at the signal, the EEG signal that you're looking for is 10 to 100 microvolts. That's what you want to see. But then once you stick the EEG in the scanner, you end up with some gradient artifacts. That's on the order of millivolts, so it's an order of magnitude higher. And you also get, these are artifacts with the interaction of the heart and the heartbeats and the magnetic fields. So those are also there. And that's on top of what you would normally have because the person is alive and they're blinking and moving and their heart is beating and also they're twitching their muscles, right? So these are basically going to be the same as what you would get if you measure EEG outside the scanner, and there are ways of dealing with that. So where are the artifacts coming from in the scanner, and what are you going to do about it? So this is a bit of a complicated plot, but really what I want you to take away from it is that when the imaging gradients are on, basically you get the artifact. And so this is basically coming from some basic physics, that if you have a switching gradient, this gives you an increase in voltage, and so that's basically what the EEG electrodes are measuring, right? Some electrical signals. So luckily for us, though, the artifact is consistent across every slice. So you can see that as you're actually imaging here, you're creating your MR image, you've got massive fluctuations here, but it's all the same for every slice. So you can actually then think of, well, if it's the same for every slice, if I know where my slice is, I can then make a template of what's happening and then subtract it out. So that's only true if your data acquisition is synchronized. If it's not synchronized correctly, and this is basically synchronization to better than a millisecond, if you don't synchronize it, you'll end up with some residuals, some residual noise after subtracting off the template. If it is synchronized, you end up getting a much, much cleaner signal. So just to look at an actual example, you can see this is data before cleaning on both sides of the same electrode set, and this is data afterwards. So it's a lot less noisy, and you're removing all of the imaging gradient artifact, which is basically drowning out all of these more subtle effects. And down here you can actually see the cardiac signal trace. It's sort of on top of one another, but hopefully you can see that. So now that we can do a pretty good job at removing the gradient artifacts. What about the BCG artifacts or ballistocardiographic artifacts? And these artifacts are very repeatable. They have a particular pattern. You can see here. So these are each of the heartbeats, and you can see it gives you basically a repeatable squiggle for each of them. And they basically come from motion that's related to cardiac activity. It's sort of maybe a bit unclear as to whether it's just the bolus motion of the ejection parts of the cardiac cycle, or if it has something to do with a little bit of head pulsation. Regardless of that, the artifact itself is reasonably consistent, and it's matched with the heartbeats with a consistent delay. So they're distributed a little bit all over the place in the brain. But again, you can actually you can find a template of them. You average all of the templates together because you lock it with the heartbeat. You can then subtract them, and then you actually can correct them pretty well. This is sort of an example. You can see that there are squiggles in the black in the background, and then you can remove them as in the red. So, of course, if you're going to do temporalate subtraction, if you're going to average a bunch of things together and then subtract them, that means the person has to remain still. If they don't remain still, you basically do end up having a few artifacts that are left behind. I mean, there's not much to say about that other than, motion is a problem in pretty much every acquisition. So try to keep your subjects still. So let me just give you an example of how this has been used, together with fMRI, with the drug. In this particular case, caffeine. I think everybody probably enjoys caffeine for the most part. In terms of the bold response, it's a vasoconstrictor. So it'll increase, I think, your bold signal. Here what they were doing was giving people caffeine and also having them do a simple visual task or something that's a little more complicated, which is just detecting a slightly different auditory tone. They did this both with fMRI and also simultaneous EEG, and they basically find very few regions of change, maybe sort of slightly in the visual cortex, but in terms of in visual cortex in terms of bold. But if they looked at the just visually evoked potentials, you can see that all of the different conditions, both caffeine and placebo and baseline, they all overlap really nicely together. So that means that, in terms of the visual task, you're not getting very much of an improvement with caffeine. But with the more complicated task, you find one small difference up in the frontal lobe, but you get a reasonably, you get a shifted peak in the EEG response. So this way you can basically try to tease out the differences between where you're getting sort of vascular changes maybe, and maybe not fully neural responses, and also get the neural responses at the same time. So if you remember back to our simulated example, if you didn't believe that the multi-echo was actually working to actually pull out this slowed task, it's very convenient that the flickering checkerboard and the contrast sensitivity is actually reflected in the EEG amplitude. So if you change the contrast, you end up with a different amplitude in the EEG. Same thing, if you basically also then detect that at one specific frequency. So you can imagine modulating the contrast and just detecting the envelope of that change. And then you can do the same task in fMRI and EEG and bridge basically the difference between the fMRI being really slow and the EEG being very sensitive and very fast. So you can have a fast stimulus, but also image it the same way in both modalities. So if you do this at the same time, so show them, do simultaneous EEG and the multi-echo fMRI, you can see that it works pretty well for the block task. And you can see that it's reasonably convincing that you're actually getting both signals agree pretty well for both of the two different RAM tasks as well. And the particular frequency of stimulation is right nicely in the visual cortex and doesn't overlap with any of the other standard things that you were measuring with EEG. So you could confirm that this kind of ramp is nicely visible in multi-echo and it's not just a spurious finding. So I think you're going to get more examples of these kinds of things in the next talks. And so I think I'll stop there. But please come to these two where I think they'll talk a little bit more about the combination of EEG, fMRI, and the things you can do with it. And with that, I think just summarize what I've talked about, and it's just that pharmacological fMRI has the potential to have a lot of benefits, but it's complicated just because you're not measuring the neural activity directly. So you need to have maybe an adjunct if you're looking for the neural signal specifically. EEG fMRI is an example of the way you can do that. There are also other things like ASL if you want to look at just blood flow or volume, and PET if you want to look at the neural bindings. But yeah, I mean, in terms of actual drug development, it's pretty exciting. It could actually have a pretty big impact in bringing new CNS drugs to the market in the future. So with that, thanks for your attention. If you have any questions, I can try to answer them. Or if you want, you can just come and ask me questions instead of having to ask questions as a group. It's fine too. Hendrik.
So, what's missing? What are you working on? What needs to be developed next? Sounds like the system is working. So, what are the remaining challenges?
Well, IRB.
Technical. Oh.
Okay. Just recently, like yesterday, basically, we did run an experiment where we were giving an injection of ketamine in the scanner at the same time with doing simultaneous EEG and fMRI. So those kinds of things have been worked out. So you can do them, and now it's a matter of just seeing whether or not, because ketamine is a glutamatergic modulator, glutamate is involved in neural signaling and so on. And so now it's a matter of whether or not you can actually pick up those changes effectively. And in some ways, if you don't actually care whether you're measuring a neural signal or not, you just want a surrogate of the drug effect, you can just go with the bold signal as long as it's repeatable, right? So you don't have to link it back to the neural signal if all you're looking for is a marker of drug action.
So EEG, if I understand correctly, is most sensitive to these large sort of somatosensory evoked potentials. In the context of drug effects of, say, depression drugs or something like that, what kind of EEG signatures?
The gamma band. So they've shown that ketamine modulates the gamma band, basically, which may or may not be perfectly measurable in the scanner because of where it overlaps with some of the artifacts. But they've basically showed that some of the bands are actually modulated nicely.
So you mentioned ASL several times, but it seems like it's not being used that much. Is there a particular reason? Because even though it has a lot of potential to use some physiologic information about the physiological parameters.
Yeah, some of the studies do try and do ASL in parallel with fMRI, but fMRI seems to be just much more popular to use. Easier to do, yeah. But it is being used. It's just not as common.
So, but if you had a good ASL protocol,
oh yeah, if there was a good ASL, yeah, absolutely. Because if they're interested in, yeah, for sure.
Anyone doing EEG and ASL?
Well, I'm not. Probably not. It depends on whether the ASL has multiple more RF pulses. Maybe you have to be careful. You're not causing SAR issues and so on. Most of the equipment is rated for one stimulus per volume, or one excitatory pulse per volume, and 3T. Although some people have higher field strength stuff at 7T and so on. But they've had to specifically test out their equipment to make it work.
Is there equipment that could simultaneously measure PET and fMRI?
Yeah, so we have NIH has PET fMRI scanner. The main trick there is that the coil, for example, since it's between the person's head and the detectors, that has to be modeled properly because if the PET measures the basically radioactivity that's coming off, right, the emissions, and so you're going to get a reduced amount from the coil, so that has to be modeled. But yes, that can be done and can be done here too at NIH. So that's something that I'm interested in doing as well, as long as you have the binding agent to bind to what you're looking for. All right, thanks.